So to go ahead and see that in this diagram mapped out, where we have mitosis and meiosis, and the starting point of the two processes, I can actually see the exact same cell being used. I have a diploid in humans, that's 2n equals 46 total chromosomes. The same here with meiosis, it's the same starting point. The difference between the two is your endpoint. That's the ultimate difference, where in mitosis, we end up getting two genetically identical daughter cells. There's that diploid 46 number again. Meiosis actually occurs in two parts. There's part 1 and part 2, and ultimately at the end of meiosis, we get 4 non-identical daughter cells. They're going to be different, and they're going to be haploid. You've halved the genetic material. Let's go ahead and discuss how that happens. Okay, let's go on and continue on with meiosis and see what's really going on with those chromosomes, how meiosis differs from mitosis, how we're halving that genetic material. Okay, and you're really going to see through the process of meiosis that meiosis I is quite different. It's meiosis II that's very similar to mitosis. In fact, meiosis II, we call this part the mitosis of meiosis. Let's go ahead and see why. And I'm going to show you guys this in different images and diagrams. Okay, so it looks like we have the first step being S phase of interphase, where chromosomes duplicate. That doesn't change. This is occurring in interphase before the process of meiosis occurs. So here I have a pair of homologous chromosomes. Homologous chromosomes are now being mentioned because they are very important in meiosis. So the first step is that chromosomes duplicate, and I can actually see sister chromatids appearing for those chromosomes. Here's that red chromosome now showing sister chromatids. It is a duplicated chromosome. Here's that blue chromosome showing sister chromatids. Now, our second step. Homologous chromosomes separate from each other. So they lose their partner. I can actually see in meiosis I, this is occurring. So the whole point of meiosis I is homologous chromosomes separating. This didn't happen in mitosis. So already in meiosis I, the end result is halving the genetic material. I'm going to get a haploid cell here. I'm going to get a haploid cell here. We get 2 haploid cells at the end of meiosis I. I want to reinforce that homologous chromosomes are separating, so they're losing their partner. Red had a partner, blue, at the beginning. This was a pair. Now, red has separated from blue, and vice versa. You've separated them. They've lost their partner. You've made what was diploid into now haploid. Additionally, appreciate that the sister chromatids on the red chromosomes have stayed intact. We have not separated the sister chromatids. The same with the blue. So the chromosomes are still duplicated. They've lost their partner. The homologous chromosomes have separated at the end of meiosis I. In meiosis II, it is called the mitosis of meiosis because in this process, it's the sister chromatids that are separating. Recap that in mitosis, sister chromatids separate. Well, that's what's going on in meiosis II. Sister chromatids are separating. I can see here, if I just look at the red chromosome, that each sister chromatid now separates into its respective cell at the very end of this phase. Same thing with the blue. And so the end result of meiosis after parts 1 and 2 have occurred is 4 non-identical haploid daughter cells. What I mean by non-identical is that they're not genetically similar. They don't have to be if we can appreciate that these blue sister chromatids are going to look a lot different from these red sister chromatids that are now separated. Now they're their own chromosome. And they're haploid and that the genetic material has halved. Let's actually look at this in more detail because we have phases occurring during each part of meiosis. So that image I showed you before is a nice, beautiful diagram showing you the overall picture of what's going on in the two different parts of meiosis and your end result. Now let's just look at meiosis I. So in meiosis I, we're going to see PMAT phases that you've seen before: prophase, metaphase, anaphase, telophase, and very similar things that are going on that we've seen with mitosis. So in prophase I, this is meiosis part 1, prophase I, it looks like I have a diploid cell starting out with, and that diploid cell duplicated its genetic material, so I have sister chromatids. I can actually appreciate those chromosomes here. There's a pair of homologous chromosomes showing sister chromatids. There's a pair of homologous chromosomes showing its sister chromatids. In prophase I, we actually have homologous chromosomes finding its partner, kind of finding and making its way towards its similar chromosome, so finding its pair, kind of lining up right next to it, because what happens is an exchange of genetic material. This is called crossing over in prophase I. I can actually see that occurring a little bit more beautifully in this image down here. So if I focus on those pink chromosomes, finding its partner, there's its partner over here, these purple chromosomes, they line up next to each other. And here I can actually see gene A, B, and C. Let me get rid of this as best I can. Okay, there's genes A, B, and C. What's going on when they're kind of finding its partner and standing next to its partner during prophase I is an event called crossing over, where genetic material becomes exchanged between the two pairs. And I can appreciate that exchange, crossing over, has occurred here, where a little bit of that purple chromosome became part of the pink, a little bit of that pink chromosome became part of the purple. So now, if we look at what was at the beginning, purely pink, purely purple, there was an exchange of genetic material that occurred. This is called crossing over. It occurs during prophase I of meiosis. In metaphase, I do also want to point out that we have our centrosomes showing that they're migrating to opposite poles of the cell. We see the nuclear envelope dissipating. This is all synonymous with what we've already learned with what occurs in prophase. The difference here is crossing over with those homologous chromosomes. Now metaphase I is going to take place after prophase I, and very similar in that we see our spindle apparatus being formed, kind of finding its way over to those centromeres of the chromosomes, and we see an imaginary metaphyseal plate occurring where those chromosomes line up. Now, the way that they line up is going to be the homologous pairs of chromosomes lining up with each other, instead of seeing one by one in a line like we saw with mitosis. And so here I can see crossing over has already occurred. Here I can see that pink chromosome lining up with its partner, its homologous pair, the purple chromosome. And then that continues on for however many homologous pairs we have. In anaphase I of meiosis, homologous chromosomes separate. So there's that spindle apparatus attaching to the centromeres of chromosomes, and I can see that the sister chromatids have not separated. They stay intact, but the pairs, the homologous chromosomes, are now migrating to opposite poles. That occurs during anaphase I. In telophase I and cytokinesis I, this image is showing both, we actually see the cells forming the end result, where now we have homologous chromosomes separated at different poles of the cell. I start seeing the nuclear envelope forming again around those homologous chromosomes to end that process. And then in cytokinesis, this is an animal cell that I'm showing you guys because I see that pinching effect occurring. That's called a cleavage furrow in animal cells, where we actually get cell splitting. The cytoplasm is now going to be splitting, breaking off until we end up forming completely whole new cells. We can appreciate that at the end of meiosis I, we get two end results, two daughter cells that are now haploid. We halved the genetic material, and they are genetically different from the starting point. We still have those sister chromatids attached. We still have that duplicated DNA. The sister chromatids are attached at the centromere. We're going to go ahead and split those sister chromatids. That occurs in the process of meiosis II. And so now we have PMET going on again. This is very much the mitosis from meiosis, where we end up getting our final result. Prophase 2 happens. In prophase 2, remember that we ended up with two non-identical haploid daughter cells. We're going to go ahead and continue on. I'm going to focus on the one on the top here as I talk through this. And so here in prophase, we have the centrosomes migrating to opposite ends of the pole of the cell. We have the spindle apparatus forming. We have the nuclear envelope dissipating. We have that genetic material kind of getting itself ready for this process. Metaphase 2 now. Metaphyseal plate aligning, chromosomes lining up. I can see that there's no longer a partner for those chromosomes that is lining up with it. It's just chromosomes lining up individually along that plate. In anaphase 2, spindle fibers attached to the centrosome, the kinetochore in particular, that protein, in order to split up those sister chromatids. I can actually visually see those sister chromatids now migrating to opposite ends of the cell, the different poles. Telophase 2, cytokinesis 2, I now have my end product where I have the genetic material on different poles of the cell. I see that nuclear envelope forming again around that genetic material. And cytokinesis 2, I see cleavage furrow formation for cell splitting to end up giving me 1, 2, 3, 4 non-identical daughter cells. These cells are going to go on and become gametes, whether they're eggs, whether they're sperm. I like this image, appreciate that genetically non-identical. So here we have a pink chromosome and a purple chromosome. Here I have a little bit of purple along with majority pink, and then a little bit of pink along with a little bit of purple here. Here I have, opposite token, a little bit of pink, a lot of purple. Just appreciate the different colors, because what that's showing me is that because of the process of crossing over that occurred during prophase 1, I now have genetically different cells resulting. Not one is similar to the other. They don't have to be. Meiosis gives us this genetic diversity.